Okay, good morning, everyone. So today will be your fifth tutorial for comparative genomics. Let me share the slides with you. Right. Okay, maybe we'll wait a few minutes. We still have a few more students to join. Right, so let's start today. Okay, so today, as I mentioned, it's going to be your fifth tutorial. It is basically a theory lecture. And today's lecture is going to be about complex evolutionary mechanisms and the different mechanisms that contribute to these complex evolutionary scenarios. I've shown a picture here which is adapted from an article, which gives an example of different kinds of evolutionary scenarios you may be able to observe when you do a comparative genomics analysis. So usually you just see something like the first, which is just speciation, where due to a speciation event, you get multiple orthologs. But there are cases where a lot of different mechanisms occur, such as lineage-specific expansion, lineage-specific gene loss, lateral gene transfer. So these kind of other mechanisms actually contribute to different kinds of phylogenetic analysis, phylogenetic tree, like seen here, right? So let's move on to today's lecture. Right, so the interplay of speciation events, which leads to divergence of orthologs, and also duplications, giving rise to paralogous families, results in complex evolutionary scenarios. So if you're just looking at just one event, just speciation, you'll see something very simple like this, right? But if you have multiple speciation event and duplication event, you will start to have very complex looking evolutionary trees. So for instance, when duplication precedes speciation, which means duplication happens before speciation, each of the paralogs will give rise to distinct linear of orthologous descent. And conversely, if duplication event happens after a particular speciation event in one lineage or in both lineages independently, a situation ensues whereby a one-to-one orthologous relationship cannot be delineated in principle. So this is an example when duplication occurs after a speciation event. So you can see speciation happened first. So from one common ancestor, it diverged to three different species, sorry. And from that, duplication event happened. So if you see the first gene set, right, two paralogs were formed. In the second gene set, there was no duplication event. And in the third gene set, again, duplication event happened and three paralogs were formed. So these three gene sets from the first family, from the not first family, first species, second species, and the third species, all these gene sets are actually known as paralogs and can be considered co-orthologous because they came from a common ancestor.
As I'm not big.
Sorry? Okay, right. So let's move on. Right, so instead, all one can say is that family AB in lineage 1, which is this lineage 1, A is in the first gene and B is the second gene, which are paralogs each other, is actually orthologous to family ABC in lineage 3. So this is gene A, gene B, and gene C. So they are orthologous, and so they are also known as co-orthologs because they come from a common ancestor, right? So this is an example of a complex evolutionary scenario that you can observe when you do comparative genomics analysis. Okay. Moving on, relationships between homologs could become particularly tricky if some of these genes in certain lineage have been lost during evolution. So if certain genes are lost, due to adaptation or environmental challenges, if it's lost, then it will be even more difficult to pinpoint what happened, or it'll be more difficult to explain what has happened, right? So these are some of the challenges that one face when you're doing a comparative genome analysis. But of course, more in-depth reading and more genome sequencing can help unravel what's exactly happening. So which means you will need to have complete genome sequences, not partial, right? Complete genome sequences. You also require genome sequences of closely related species or closely related families in order to really explain how things or how an organism have evolved over the years, right? So reliable identification of orthologs is only possible when you have complete sets of genes from two or more genomes to compare. So if you're going to compare genomes and you have one which is just partially sequenced, maybe you are supposed to have a 6 million base genome, right? But your sequencing results are not sufficient and you only get about 4 or 5 million bases. So 1 million base is actually missing from your genome. So if you use that sort of partially sequenced genome to do a comparison, it is quite difficult to derive accurate conclusion, right? It's because you may never know if there is supposed to be an orthologous gene in the 1 million base which is missing, right? So this is what it explains here. Indeed, if one of the compared genomes, let's say you're comparing two genomes, right? And one of it is actually incomplete or partially sequenced, you are expecting both to be 6 million bases, but one is only 4 million, the other one is 6 million bases. So a possibility always remains that the true ortholog or true orthologous gene is hiding in the unsequenced part. But of course, you cannot confirm that it is whether it's present or absent until you sequence or you get a complete genome sequence of the second genome, right? Even with complete genomes, sometimes identification of orthologous gene sets is not as simple as we expect it to be because of the complex evolutionary scenarios. So for instance, if a gene has been lost, it is difficult to identify this or to realize this unless you have done genome sequencing of a lot of closely related species or closely related genus to derive to a conclusion that, okay, the gene was there before, but it has been lost, right? In principle, complete phylogenetic analysis of all groups of homologous genes is required to decipher true orthologous relationships. So this is what I meant. So if you really want to study very, in-depth, do a really in-depth phylogenetic analysis, it is actually very important to analyze a lot of groups of homologous genes. So you cannot just compare just a few genomes, maybe 4 or 5, to be able to derive to a very accurate understanding of what has happened. Okay, but of course, for comparative genomics, the cost, I'm not expecting you all to sit and analyze a few hundreds of genomes. That's not possible with the computing facility we have. In fact, yeah, it's not going to be possible, and it will take a really long time, right? And given only a semester, it will be impossible for you to finish this, right? So some of them actually do comparative genome analysis for their PhD projects. So there you can actually sit and compare hundreds of genomes and come to a really nice conclusion. But having just one semester, it is quite impossible. So that's why we are sticking to smaller genome sets to be able to do some analysis to give you an idea what you can do given a few genomes, right? So this is, as I mentioned, an extremely labor-intensive task. Moreover, it is well known that not all phylogenetic trees provide the required resolution. So if you don't have complete genome sequences, if you don't have, for instance, genomes of closely related species or genus, it is going to be a little tricky to be able to explain what exactly has happened in the evolutionary tree, right? Okay, so moving on, these are some of the patterns and mechanisms in genome evolution, um, some of the significant events that occur during genome evolution, right? So we will look at these 5 and also what these mean and how do they look, and to give you an idea of how evolution actually happens, right? So these are some of the mechanisms that contribute to the complex scenarios that you see. So the first is genome arrangement, lineage-specific expansion, lineage-specific gene loss, lateral gene transfer, and the last one is non-orthologous gene displacement. So among these, I think you must be familiar with one term, lateral gene transfer or horizontal gene transfer. So can y'all explain or guess what this is, or give an example of how a horizontal gene transfer occurs? Anyone? Are you all there? But so many participants, not one person is speaking. So I'll pick one. All right, Kale?
Yes, doctor.
Yeah, so can you give an example of a lateral gene transfer?
Like
Yeah.
unicellular organisms.
Uh-huh. How do they transfer the gene to another organism? You remember, I think you must have studied this in either genetics or genetic engineering. Anyone else? Right, okay. So let's move on. Do you remember the terms transduction, conjugation, transformation?
Yes, doctor.
Yeah, so these are some of the examples of how lateral gene transfer occurs. So we'll move on. So the first one is genome rearrangement. This is also known as evolution of gene order. So in what researchers notice is when they do a comparison of genomes, they notice that although the nucleotide sequence is sort of or quite conserved, or protein sequences are quite conserved among closely related species or genus, they notice that there's quite frequently rearrangements in the order of the gene. Okay, so for instance, over here, there's an example in this picture. So the bottommost is a chromosome from human DNA, right? So you can see gene 1, 2, 3, and all the way to gene 11. And if you see the order in a mouse, the same genes in a mouse, right? The order is actually flipped and rearranged. So the first gene is okay, it's in the correct same order. And then the second gene, which was supposed to be number 2, it's sort of like flipped, or there's a change, and you see gene number 7 is in the second position, right? And it is -7 because it is flipped in the other direction, right? You can see the arrow is in the opposite direction. Okay. Next, in the third position, you have gene 6 for mouse, and in human is the third gene. And the fourth position, you see gene number 10 in the reverse order, right? The fifth position, you see gene number 9 in the same order. So you can see that the sequences are like the sequence itself, the nucleotide sequence or the protein sequence itself may be very conserved or very minimal mutations or changes. But the order of these genes which are present are completely rearranged, right? So this is quite frequently seen even among closely related species. Could be even bacteria or eukaryotic genes, right? So, so this is an example of genomes of the closely related bacteria, Mycoplasma genitalium and Mycoplasma pneumonia. For example, their genome actually consists of 6 large segments with similar organization of genes. But the segments themselves are shifted relative to each other and partially scrambled in the 2 genomes. So you can see in the x-axis over here, you have Mycoplasma pneumonia strain genome, right? And this x-axis actually represents position 1, right, at the 0 coordinate, right? It's actually position 1 in the genome of Mycoplasma pneumonia, and all the way until position, let's say it has a 4 million base genome. The last position here is the 4th million base, right? And similarly, in the y-axis represents Mycoplasma genitalium's genome sequence, right? So position 1 is at the coordinate 0, 0, and all the way, let's say again 4 million bases, the genome is 4 million bases. The 4th million base is at the end of the y-axis, right? So each time they see, okay, let's say the first base is adenine in Mycoplasma pneumonia, and it's the same in Mycoplasma genitalium. It's also A. So you actually draw a dot, right? Similarly, in the second position, you see, let's say T in thymine in your Mycoplasma pneumonia strain, and you see the same base in Mycoplasma genitalium genome in the second position. So you draw a second dot. So that's how you actually draw this plot. This is known as a gene dot plot. Okay, so each time you see the same base present in the coordinate, present in genome, the genomes you're comparing, like over here is comparing these 2 genomes. Each time you see the same base, you draw a dot, right? And if it's not the same, then you don't draw a dot. So you can see finally it comes to about 6 different segments, right? So this segment, the first segment you see here, they are extremely similar or synthetic in Mycoplasma pneumonia and Mycoplasma genitalium. So maybe in the first, maybe 100,000 bases, you see a very similar nucleotide sequence, and arrangement of gene. Then you see a very big gap here, like a break, like nothing is happening there. So basically there is no similarity in these two genomes, right? It's not that nothing is present there, there is, but there's no similarity. And here again, there are a few, lines, diagonal lines, but it is not like a collinear line. So which means there are dissimilarities in the position of the gene, right? In Mycoplasma pneumonia strain, it is around, for instance here, and in Mycoplasma genitalium, it's somewhere here. So you don't see that it's exactly similar. So with this, they actually did a comparison of these two genomes, and they found 6 different segments. This is the first, right? And this is the second segment. This is the third segment, fourth, right? Fifth, and a sixth segment. So in these 6 segments, they found that they are very similar in the arrangement of genes. Okay, and they actually did many different comparisons, right? Genome comparisons, and they found that, for instance, when they compared Haemophilus influenza genome with E. coli's genome, they found a lot more differences. Of course, because they are not, related; they are quite distant. So they found a lot more rearrangements done, or even between E. coli K12 and its pathogenic relative E. coli 0157. So even, between the same species but different strains, like one is a pathogenic strain and the other one is the non-pathogenic strain, they found that there are, dissimilarities in terms of arrangement of the genes in the genome. So a gene order comparison plot, or a dot plot, so this is what it is very useful to study similarities in genome organization between two organisms. Okay, so a gene order dot plot, or also known as gene order comparison plot, is basically a 2D plot, right? This is the 2D plot I'm referring to. So this is usually used for comparison of two genomes from different taxa. That means can be from 2 different species, 2 different genus, 2 different families. It depends how you want to look at it. But of course, if you're going to choose genomes from completely different families, then you will have a very confusing looking plot. Yeah, I'll show you an example after this. Right, so normally researchers do comparisons of these genomes to have an idea of, to look at synteny regions, right? So synteny, the term, is defined as 2 or more genomic regions that are derived from a common ancestral genomic region. Okay, so in this case, like you can see, this is a synthenic region. So you can be quite confident that this region came from a common ancestor, like they share a common ancestor. Right, so the evidence for synteny is the identification of a set of homologous genes in 2 genomes that have a collinear arrangement. So this is what we mean by collinear arrangement, where if it's exactly the same, the gene order, you will get a very nice straight diagonal line, right? And this is not too bad. You can see that there is sort of a diagonal line with a bit of breaks a few places, but almost similar arrangement in genes. Okay. When such a pattern of gene order conservation is discovered, the most suitable explanation is that the 2 regions are actually related through a common ancestor. So when you see that the regions, the genes are actually, arranged in a similar order, you can quite confidently say that they are most likely related through a common ancestor, and hence they still have, or they still maintain the gene order arrangement. While synthenic dot plots are useful for identifying these, synthenic genomic regions, they are also useful for identifying genomic regions that have undergone an evolutionary change in one of the 2 genomes being compared. So over here, you can see that, they are very similar, right? But you can also look or you can also identify if there is, some changes in the genome. For instance, if there's an insertion or deletion or a gene transfer or inversions, this can also be observed. So I will show you examples after this, right? So this is just to give you an idea of how to interpret a dot plot. Again, this is an example of the same thing, a dot plot. But here, the genomes being compared is an E. coli strain, REL606, and another E. coli strain, DH10B, right? So one x-axis is REL606, and the y-axis is E. coli DH10B. And you can see this is also, again, a almost collinear arrangement. So you have like an almost 45, almost a perfect diagonal line that you can see here. Okay. So each axis of the dot plot is in nucleotide units, as I mentioned. So the first position is of REL606 nucleotide is here, or it's also known as ORI, or origin of replication, right? And it represents one of the two genomes laid end to end. So let's say you have a 4 million base genome. You basically have an x-axis where you have base 1, base 2, base 3, all the way to base 4 million, right? So the lower left, which is your 0, 0 coordinate, actually represents the start of each genome, or also known as ORI. So if you're referring to bacterial genomes, you only have one origin of replication, because it's a circular genome. So you have one ORI, and then you start basically drawing dots each time you see an exactly same nucleotide being present in the other genome, right? So each putative homologous gene pair is drawn as a gray dot. Of course, you can't see the gray dot here, okay, on the dot plot, with its X and Y position corresponding to the genomic position of each gene in their respective genomes. So when they finally keep drawing the gray dots, okay, they come up with these green lines, which are gene pairs that have been inferred as synthetic or similar. So every time they see a region which is exactly, similar or synthetic, that area becomes a green zone, right, or green region. So this entire area, for instance, the first, for instance, this green line basically means a few genes are in the same order in both REL606 strain and in DH10B strain, right? So the collection of these dots appear as a green line, which for the comparison of these two genomes results in a nearly continuous green line running 45 degrees up the plot. So you can see that an almost continuous line is formed. So you kind of can interpret that they have a very similar gene arrangement in both the genomes, right? So this means that these genomes are completely synthetic. That means completely similar, and so they share a common ancestor. But from the name, you can see they are both from the same species, E. coli strain, E. coli. They are just different strains, right? So this is one interpretation you can do. But as you can see that there are some arrows here with numbers, right? And the regions that they're actually showing are breaks, right? So there are like certain regions where there is nothing, no lines are being formed, right? Okay, so this is what I said. They are also known as breaks or discontinuities between them. So these breaks in the synthetic path between these genomes are due to genomic changes happening at a larger scale than a single nucleotide polymorphism. So it's not just one base difference. You see actually probably about hundreds of bases difference. And so you see that there is no dots in the region. So it's like empty, or there's a break. And these are most likely due to insertions in one of the genomes, or deletions in the other genome of many nucleotide chunks in these genomes, right? So it's not just one or two bases, it's like multiple nucleotide chunks are actually either inserted in one or deleted in the other. So that's when you see these breaks in the genome. So in order to accurately account and characterize what's exactly happening there, why there's a discontinuity in the dot plot, you need to perform a higher resolution analysis of these regions. And there is a software which also helps to do this. So the software is known as GBO, but there are many other softwares available. This is I think quite commonly used and quite simple to use. So you can, after the class, go and have a look at this software, right? It's not a Linux one. It's just a web server based one. So all of y'all can go and have a look, right? So you just have to search Jibo, and you'll be able to play around. I can show you this Thursday how it's done, but for now I'm just going to discuss how it looks, right? So using the GIVO analysis results, you can actually characterize the differences between these two genomes that you've chosen to compare at these synthetic regions. So for instance, over here, you're comparing the REL 606 with K12, DH10B strain. So for instance, the first panel, right? The first panel is actually the REL 606 genome. And you can see two lines here, right? And there's one dotted line in the middle. So this dotted line is basically, separating the top and bottom strands, the forward strand and the reverse strand of the REL 606 genome, right? So in the forward strand, you can see these green blocks. These green blocks actually refer to the genes which are present in there. And if you want to know what gene it is, you just have to place your cursor there, and it will actually give you an annotation of what gene it is and what the gene ID is given, what product or what protein product the gene actually codes for, the length of the gene, the position or location. So it's actually in this position, 941,285 bases. So these are the information that's given when you just, basically just run your cursor over the gene, right? And this, after the separating line, you see again green blocks. These are genes which are present in the reverse strand of REL 606 strain. Similarly, this is a second panel, right? And this second panel refers to DH10B genome, right? Here also you can see a dotted line. So the top, the blocks which are above the dotted line are genes which are present in the forward strand of the DH10B genome, and the blocks, of the green blocks at the below the dashed line is the genes present in the reverse strand of this genome, right? So these pink regions, right, that you see over here, like a long line, long block, and this long block. So these are actually, you can see that they actually show an overlapping region in this genome. So this is actually referring to synthetic regions, right? So this long red block here actually has a synthetic region or similar region in the genome, in the DH10B genome, and this is where it's present, right? So these two regions are exactly the same, meaning the same genes are present in these two genomes, right? Although the same genes are present, but you can see some are actually the gene blocks, some are actually present in the reverse strand in one genome, and some in the forward strand of the genome, right? Similarly, you can see a second region of synthetic block here. So this entire pink region actually overlaps perfectly with this part of the DH10B genome. So the genes which are present in this region, right, of REL 606 strain is also present in DH10B strain. But another interesting thing you can see is the gene order is exactly the same. Like you see in the forward region, you have all these genes. You can see the same genes are actually present in the DH10B strain, right? Similarly, even in this region, right, the second block, if you see this area, it's exactly in the same order in the second genome, right? So this is how you see synthetic blocks, or also known as similar regions in two different genomes, right? And over here, you can see there is one region of, right, the genome of 606, REL 606. There is no color. You don't see a red block over it. And that region is actually not present in the second genome that you're looking at, DH10B, if you can see. Right, so which means most likely these genes are actually deleted or not present in the DH10B genome, right? And are only present in the 606 genome. So this is where you see breaks in your dot plot, right? So you see these breaks where there's no lines at all. So this is, if you look closer into the genome, you can see why this happens, because basically these genes are not present in the DH10B, hence there is no synthetic region, and you cannot draw dots or lines in your dot plot, right? So this is an example of how you see when there is an insertion or a deletion of one or many genes present in a genome, right? So you can do this by doing a gene order dot plot or gene dot plot analysis. And to really understand what's happening, like when you see a break, you need to do a higher resolution analysis to see what genes may or may not be present in that area, right? So you can use this software called Givo to do this. Okay. Any questions so far? Are you able to follow?
Yes, Miss.
Yes, doctor.
Okay, so I'll move on. Moving on to again just genome order rearrangement is right. So you're looking at in the chlamydial genomes here. So these are actually from the same genus, right? You can see a genome scale alignment is readily traceable along the main diagonal line, although gaps in the alignment and two major inversions are equally obvious. So the first plot shown here is actually a comparison between Chlamydia trachomatic, which is in your x-axis, versus Chlamydophila pneumoniae, which is in your y-axis. And you can see that they are diagonal, but in the opposite direction, right? So, and these regions where you see, lines in the opposite direction, these are actually where inversions are happening, inversions in the order of the gene, okay? So in contrast, when you're doing comparison of genomes from completely disordered, I mean, completely different family or different genus, you can see a very confusing or complex looking dot plot. So here in the second dot plot, it shows comparison of E. coli and Pseudomonas aeruginosa genome. Okay, so they're from two different genus, but they're both gamma proteobacteria. So you can see the dots are all over. You don't really see a nice line or a particular pattern or trend. It's just everywhere scattered. So this means that there is, almost no similar arrangement or synthetic regions in the genome. You may have the same genes in both they definitely have the same genes, but the order is completely a chaos. It's not in the same order, right? So that's why you get a very complex looking dot plot. And this is normally obvious when this is what you normally see when you do a dot plot when you're comparing genomes from quite distantly related organisms. For instance, if you're trying to look at similarities between a bacterial genome and an archaea genome, you may also have a dot plot that looks like this, right? Right. So on a smaller scale, however, there is also always an important conservation of gene order within operons. So I'm quite sure you have heard of this term operon before. Basically, a few structural genes are placed together because they probably work in the same pathway or metabolic mechanism. So they are all controlled by one promoter and one operator. Okay. So normally gene order in an operon does not change, right? They are normally conserved. You still see like let's say gene XYZ is present after the operator, usually the order of these genes will be conserved across all genus that have the same operon, right? The order of the operon itself, the whole operon itself might be different, but the genes within the operon will not, the order of the genes within the operon will not be different, right? So extensive genome comparisons have shown that in each genome, about 5 to 25% of the genes actually belong to conserved operons, which are basically strings of genes that are shared with at least one relatively distant genome. So a few operons that are conserved in distantly related prokaryotes actually consist of genes for ribosomal proteins and some other components of the translation machinery, right? So moving on to the second mechanism, lineage-specific expansion of gene family, or also known as LSE. Okay? So LSE is actually defined as the emergence of multiple gene copies, right, either by duplication or lateral gene transfer events in a specific lineage. So genome comparisons actually suggest that this LSE of paralogous gene families, which in some cases account for a sizable fraction of a genome, is one of the major mechanisms of adaptation. So we notice this especially when organisms are trying to adapt to a particular stress or environmental challenge. They usually have this lineage-specific expansion of genes happening in them. And this actually gives rise to abundant gene families. And it has been shown to play a vital role in how prokaryotes actually gain new functions and adapt to their environments. So when they have new challenges in the environment, you know, they need to adapt to have different functions to be able to survive in the environment. So in order to survive, a lot of these organisms actually expand these genes, or basically have duplication events happening. So you have multiple copies of the same gene. So one of them will maintain the original function of the gene, while the others actually can evolve to have new functions which can help them survive in the environment. So you can see over here examples of some species and the number of copies of a gene, right? So for instance, Mycobacterium tuberculosis, there's one protein family, which is known as PPE, which is basically protein glutamate protein family, all right? They have 90 copies of this gene in their genome. So basically you can expect different functions of these proteins, right? These genes actually function differently. And for instance, this PPE protein family, their likely function is as a surface antigen and interacts with host cells. So each of it may have different ways of interacting with host cells, or they may have different what do you call receptors or catalytic mechanism and interact with the host cell a bit differently. So similarly, you can see different organisms here, right? So for H. pylori, it has 34 copies of HOP protein family. Again, this is a surface antigen and interacts with host cells. So especially in parasites, right, which bacteria organisms that are living with living on or depending on a host, they normally will have expanded genes or expanded gene families in order for them to interact with the host cells, because that's what they require. They cannot make their own food, they cannot do a lot of things on their own, they depend on their host. So they try as much as possible to have many genes which are able to help them interact with the host cells. So you can see most of these are actually parasites, and you can see the number of copies some of these organisms have for certain protein families. And you see most of them are either surface antigens which interact with host cells, or some proteins which are involved in sensing environmental stimuli or signal transduction. These are all basically to help them survive, right? Right. So in pathogens, for instance, Mycobacterium tuberculosis and Helicobacter pylori, the most conspicuous expansions are those genes encoding factors involved in interactions with and survival within the host organisms. So they're very selective of which genes they want to expand, right? Something that really is required for their survival, because they are basically, depending on their host. They need these genes that interact with or survive, interact with and survival within host organisms. In contrast, if you look at genomes of free living autotrophs that, they don't depend on their host, for instance, an example is Cyanobacterius cyanobacterius species, and this A. fulgidus. Okay, this is a hyperthermophile. If you look at their genomes, the largest expansion actually involves mostly signal transduction proteins or sensor histidine kinases and related ATPases. So they require these to be able to sense their environment, and so that they can adapt to the environment to be able to survive, to make their own food and survive. So you can see that the choice of genes that these organisms decide to expand are different. If it's a parasite, they basically expand genes which are involved with host interaction. If they are free living organisms that can make their own food, they are more interested in having expanding genes which are related to sensing the environment, right? And making their own energy source, ATPases. Right, so the next mechanism, right? So the previous one we spoke about how there are lineage specific expansions. So basically they make more copies of certain genes which are very important for their survival. The next mechanism we're going to look at is lineage specific gene loss, right? So in over the years, when a particular organism doesn't require certain metabolic pathways or certain processes to survive, they eventually start dropping the genes or losing those genes because they want to have a minimal set of genes to survive, right? So many pairs of closely related organisms, we have seen that they have very different numbers of genes, right? For instance, E. coli K12, right, has 7 times more genes than a symbiont, which is a Bushnera species. So these two are actually bacteria. They're very closely related. If you look at the phylogenetic analysis, right, phylogenetic tree, E. coli and Bushnera are like side by side. Okay, but if you look at the number of genes which are present and the genome size, right? So the genome size of E. coli is about 4.6 million bases, but Bushnera is only about 641,000 bases. So it's a very small genome. And if you look at the number of proteins that they code for, Bushnera only codes for 500 plus proteins, but E. coli codes for about 4,000 plus proteins. Okay, so you can see that there is a vast difference in the number of genes present in them. The same phenomenon is also seen throughout eukaryotes. For instance, your Saccharomyces cerevisiae, for example, has about 6,000 genes, which is about 2,000 genes fewer than its relative, which is Aspergillus. So they have actually done some analysis, and they found that in some genomes, they have lost some genes, right? So gene loss has happened during over the years. So all these observations show that certain phylogenetic lineages experience a significant gene loss, and is often linked to the adaptations to parasitic lifestyle. Okay, that is one, adaptation to parasitic lifestyle, or intracellular symbiosis, or it's just because of adaptation to a constant range of environmental conditions. So, when you're adapting to a parasitic lifestyle, these organisms may not require some of the genes which is required previously to survive, right? Because they're going to be getting all their food source, everything from the host. So when that happens, they don't require the genes anymore, and just producing proteins, these genes, right? Okay, coding for these proteins is just going to waste the energy. right? So what they do is over the years, they stop or they basically lose these genes because they are no longer required for their survival, right? So one example, a very famous example, is the well-known absence of biosynthetic pathways for 12 amino acids in humans. So you remember we have essential amino acids and non-essential amino acids. So some of these amino acids, in the beginning, we were able to produce all these amino acids, right? But because humans started consuming a different kind of diet, and in our diet, we were able to obtain these amino acids. The Basically, what happened was these genes slowly was lost or was removed from our genome, right? So this was made possible by the abundance of these amino acids in the food which was consumed by their common ancestors at the time of the divergence. So an analysis of gene loss in bacterial parasites showed that in many cases, it led to also elimination of entire pathways such as amino acids, nucleotides, and cofactor biosynthetic pathways. For example, a number of parasitic bacteria actually lack pyrimidine biosynthesis genes that are present in their free living relatives. So this is mainly because they no longer require their pyrimidines to be synthesized by themselves. They're basically relying on their host for this. So there is no reason for them to keep the genes anymore. And even if they keep the genes and they start producing these proteins, it's basically a waste of energy and yeah, basically a waste of energy. So what they do is over the years, they just lose the gene in order to conserve energy and only keep those genes which are really required for their survival. So if the necessary nutrient is already available in the medium or in their food, all right, the genes responsible for its synthesis becomes redundant and can be eliminated. So that's how they start losing those genes. Moreover, once at least one of these genes are lost, expression of, for instance, if you look at a pathway, right, you may have 10 different genes which are necessary to be able to, for instance, to, to be able to create pyrimidine. You need 10 different genes which codes for 10 different protein products. But when at least one of these genes is lost, what happens is there will be an accumulation of another metabolic product, right? Because the other genes are still active and they're still producing their proteins. What happens is there's an over accumulation of a protein product. And over accumulation of any protein product, as you know, will cause damage to the cell, right? So it can turn harmful. So what happens is slowly, because of the evolutionary pressure towards called evolutionary pressure, what happens is that there's a coordinated loss of genes. So it's not just one gene is lost. Over time, all the other genes are also lost. All the other genes which are required for instance, pyrimidine biosynthesis pathway, right? So if there are 10 genes which are required for the synthesis, all 10 are lost in one go, right? So gene loss is therefore actually a driving force for changes in the genome as well as in the morphology, right? So the next one is horizontal gene transfer. This is also another mechanism which contributes to a complex evolutionary scenario. And this is I'm very sure you all are very familiar with already. So this is also known as lateral gene transfer and is very prevalent in prokaryotes, right? This can be defined as the transfer of genetic information from one genome to another. And this is exactly the opposite of vertical transfer, which is basically transfer of genes from ancestors to progeny. So that is a vertical transfer. And if you're transferring genes between from one species to another, or from one genus to another, then it's known as a lateral gene transfer or a horizontal gene transfer. And there are a few mechanisms which help, which do this. They are transformation, transduction, and conjugation. So the pictures are shown here. And I'm not going to go more detail I'm not going to elaborate on this as I expect you all to be already familiar with these terms. think you have learned this in genetics already before. So these are some of the mechanisms that that contribute to horizontal gene transfer, right? And one of it is with the help of phages. So you have bacterial transduction happening with the help of phages, where it transfers a gene to another genome, right? So genome comparisons actually have helped to uncover numerous cases of horizontal gene transfer before between organisms belonging to distinct phylogenetic lineages. Uh, Archaeal genomes presented actually a very interesting picture, with some genes having close homologs only among eukaryotes and others being very similar to bacterial homologs. So you expect archaea to have genomes which are similar to bacteria, but they actually have found genes um, which they share with eukaryotic genomes as well. So those involved in information processing, for instance, your translation, replication, and transcription, actually have shown similarity to eukaryotic genomes, while genes which are involved in metabolic enzymes or structural components seem to have a very bacterial-like structure or bacterial-like sequence. Okay, right. The last one is known as non-orthologous gene displacement. So this is also another mechanism that contributes to the complex evolutionary scenarios that we see, right? So proteins responsible for the same function in different organisms usually show significant sequence and structural conservation. If you remember from our previous class, this was what we discussed. Usually when a protein has a same function, they usually have a sequence similarity, and the structure of the protein is also very similar. However, there are certain or some exceptions to the rule, right? So apparently, even unrelated enzymes with the same function has been observed. So even the structure is different, but structure is different and they have come from completely different ancestors, but they find them having the same function, right? So an example is this enzyme known as fructose 1,6-bisphosphate aldolase, found in yeast as well as in rabbit muscle. And these two enzymes, referred to as class 1 and class 2 aldolases, were later actually shown to be associated with different phylogenetic lineages and different catalytic mechanisms, right? And they also have very little structural similarity, but they perform the same function. Okay, the way they perform is a bit different because the catalytic mechanism is different, but at the end of it, they do the same thing. So that's when they invoked this new mechanism called non-autologous gene displacement. So the definition of this is the displacement of a particular gene by an unrelated, meaning non-orthologous, but functionally analogous gene, which means they have the same function, but they don't come from they are basically non-orthologous or unrelated, right? So enzyme recruitment is a common evolutionary phenomenon leading to non-orthologous gene displacement. Typically, one of the two non-orthologous enzymes with the same catalytic activity belongs to a diverse family of enzymes and could have evolved by shifting the substrate specificity of a related but distinct enzyme. So basically, it is certain mutations that have helped in shifting the substrate specificity so that they have the same function, but the mechanism is a bit different, how they do it. So the idea of non-orthologous gene displacement was actually originally developed in conjunction with the concept of minimal gene set for a living organism. So what is this minimal gene set concept? So this is actually construed as the minimal set of genes which are required for the functioning of a modern type cell, even under the most favorable environment conditions, including when there is abundance of nutrients and absence of competition. So they actually did a comparison of two bacterial genomes. They are both parasites. Okay, they compared these two genomes to see what genes are very, very important for their survival. So when what they decided was, after doing a comparison of these two parasites' genomes, and all those genes which are present in both the genomes are basically the minimal genes which are required for survival, because they're parasites. They depend on their host, or the media they grow in has everything they require. So what is the minimal genes? They researchers basically studied their genome to see what is the minimal, or what minimum genes they require to still sustain if everything else is provided to them, right? So the assumption is these two bacteria, which belong to distant phylogenetic lineages, have been independently losing their genes during the adaptation to parasitic lifestyle. And whichever common genes which remain in both the genomes were most likely to belong to the minimal set of essential genes for their survival. It was noticed, however, that for certain essential functions, for instance, glycyl tRNA synthetase, there was no autologous pair of genes in the two bacteria. Hence, they invoked this new mechanism called non-autologous gene displacement. And non-autologous gene displacement is prominent even in the DNA replication machinery, which is a very important function system in all functional system in all the cells. All right, so that's it for today's class. Do you all have any questions? No questions? So I assume that you have understood. I will send these slides for you all to go through. And if you have any questions, just write to me. And the other thing is the bioinformatics students, I want you all to stay back. We'll have a short discussion. And the biotech students, in our next class, I hope you have already discussed with your friends, your group members, and decided on a project that you want to work with for comparative genomics. So maybe this Thursday we can have a short discussion with each group to see what your ideas are, what you want to work on, and we can discuss further. So I would like you all to just make maybe a 3-slide presentation. It's not really a presentation, it's for the sake of discussion. Just 3 slides, okay, every group. Just 3 slides. First is introduction to the project that you want to work on. All right. Second, I want you to have the rationale or justification why you want to compare these genomes. And the third is an expected outcome. Like, what are you expecting to see? Are you going to look at phylogenetic analysis, or you're going to look at certain genes that you're interested in seeing whether they're similar? So just 3 slides. One, introduction. Second one is justification, and third one is expected outcome, right? So each group, I think there are only 6 groups. Each of y'all just make a 3-slide presentation, and we shall discuss on this Thursday during our comparative genomics practical session, right? So if you have multiple ideas, you can also share that with me, and we'll see which will be the most feasible to work on, right? Any questions?
No, doctor.
No, doctor.
All right, okay. So I'll see you all Thursday for the discussion. And bioinformatics students, please stay back. The rest of y'all can leave. Thank you.
Thank you, doctor.
Thank you.
Thank you, doctor.
All right, thank you.
Thank you, doctor.
Right. Okay, so you all had a lot of questions you all wanted to discuss. Bioinfo students, are you all there?
Yes, doctor.
Right. So yeah, what was it you wanted to discuss?
Actually, we already finished our slides, but then we are not sure whether it's correct or not. Since we already have the methods and tools in the article itself, so is it okay to add the tools apart from what is in the article?
Come again. So is it okay to?
To add tools, which is not in the article actually. But
Yeah,
we wanna add some additional slides.
yeah, no problem. Basically, I want to see what it is. Basically a review. I want to see what you have reviewed and whether you got an idea of what metagenomics is, metatranscriptomics is, and what are the common tools which are used. It doesn't need to be from one particular paper. So, you, different papers, actually different researchers use different tools and there's no one right way. So what I wanted you all to do is basically a review of all the different tools which are used for the analysis. So it doesn't, you can have multiple tools doing the same thing. And it's just going to be a discussion session where we're going to look into that. So are you all actually prepared to discuss?
Yeah, just halfway, but then now I'm planning to add some of the tools. That is what I wanted to clarify.
All right, can. So maybe next Monday we can do that. Like if you'll finish your slides and we can discuss on Monday, next Monday. And the rest of y'all, I think all of y'all had some question. Ovinia?
Miss,
Yeah.
and then for the output of the analysis, what we actually need to include there, Miss?
Basically expected outcomes. So are you going to get a pathway analysis, or are you going to get like what do you say, OTU binning? So how does the output look like?
Oh, okay, Miss. Thank you.
Probably profiling of the microbes. You probably will get a PCO-A plot. So those kind of things are expected outcomes. So that's what I wanted to see. You can actually take pictures from papers just to show how the output will look like, the expected output. Okay, Bing Feng, any questions?
Miss,
Yeah.
Doctor, you need to work all the workflow, is it? Not only specific one.
So it's more like a general workflow, but if there are, maybe a few different methods for analysis, you could have a few workflows or a few flow charts. I'm okay with that as well. But usually they have a common workflow of a common method, just that they use different tools for the analysis. Okay, Prashad and Savitri?
I had the same question as Priscilla, so now I already understood it.
Okay. Savitri, any questions?
The same question as Hovinia, Miss. She already asked the question.
All right. So if you all are clear, then so for this second project, we will discuss on Monday, next Monday, and this Thursday, I would like you all to do the same thing as the other groups, where it's just 3 slides. Your introduction, justification why you're doing a comparative genome analysis of the genomes you're choosing, and also again an expected outcome. Are you going to look at phylogenetic trees, or are you going to look at synthetic blocks, those kind of things, or a SNP analysis that also can be done, right? You all are clear about the first project, right? And the first project is a bit more simpler. No metagenomics or metatranscriptomics, is basically comparative genome analysis. So just 3 slides will do. And yeah, all of y'all are in one group, right?
Yes, doctor.
Yes.
Yeah. Right. So any other questions? Are y'all able to use your virtual box? Are y'all training? Have y'all tried?
Yeah.
Right.
Yes, doctor.
Any problems?
So far no.
All right. Okay. Right. If nothing, then I will see you guys on Thursday. Thank you.
Okay, thank you, Miss.
Doctor. Thank you, doctor.
Thank you. Bye-bye.
Thank you, Doctor.